is Mary and welcome back to Brain in Rain ASMR. Today I have a new microphone and today we are going to read a book about sleep science, or to be more precise, it is a chapter in a big neuroscience book, chapter 7. It is called Sleep and Dreaming, written by Francesca Ciclari and Giulio Tononi. Anyway, let's start. Studying mental activity during sleep offers a unique opportunity to find out how changes in consciousness are associated with changes in brain activity. Indeed, sleep brings about at once the most common and the most dramatic change in consciousness that healthy subjects are likely to witness, from the near fading of all experience to the bizarre hallucinations of dreams. At the same time, the brain goes through an orderly progression of sleep stages, which can be identified by recording the electroencephalogram, EEG, eye movements, electroculogram, and muscle tone, electromyogram, and which indicate that major changes in brain activity are taking place. Within each sleep stage there are frequent, short-lasting EEG phenomena, such as slow oscillations and spindles, which indicate precise times at which brain activity undergoes important fluctuations. There are also orderly spatial changes in the activation of many brain regions, as indicated by imaging studies. All of this happens spontaneously and reliably every night. Moreover, similar changes occur in animals which have spearheaded detailed studies of their underlying neural mechanisms. This chapter will first examine how sleep is traditionally subdivided into different stages that alternate in the course of the night, and will consider the brain centers that determine whether we are asleep or awake. This chapter will then discuss how brain activity changes between sleep and wakefulness, and consider how this leads to the characteristic modifications of consciousness. That was the introduction. Sleep stages and cycles. In the course of the night, the electroencephalogram, electroculogram, and electromyogram patterns undergo coordinated changes that are used to distinguish among different sleep stages. Wakefulness. During wakefulness, the EEG is characterized by waves of low amplitude and high frequency. This kind of EEG pattern is known as low voltage fast activity, or activated, when the eyes close in preparation for sleep. EEG alpha activity, 8 to 13 Hz, becomes Non-REM sleep in turn is divided into stages N1, N2, and N3. We have a little diagram here that shows how EEG looks during wakefulness and different sleep stages. Non-REM sleep, stage 1. Falling asleep is a gradual phenomenon of progressive disconnection from the environment. Sleep is usually entered through a transitional stage, stage N1, characterized by loss of alpha activity and the appearance of low voltage, mixed frequency EEG pattern with prominent theta activity 3 to 7 Hz. Eye movements become slow and rolling, and muscle tone relaxes. Although there is a decreased awareness of sensory stimuli, a subject in stage N1 may deny that he is asleep. Motor activity may persist for a number of seconds during stage N1. Occasionally, individuals experience sudden muscle contraction, hypnic jerks, sometimes accompanied by a sense of falling and dreamlike imagery. Individuals deprived of sleep often have microsleep episodes that consist of brief 5 to 10 seconds bouts of stage N1 sleep. These episodes can have serious consequences in situations that demand constant attention, such as driving a car. Non-REM sleep, stage N2. After a few minutes in stage N1, people usually progress to stage N2 sleep. Stage N2 is heralded in the EEG by the appearance of K complexes and sleep spindles, which are especially evident over central regions. K complexes are made up of a high amplitude, negative sharp wave followed by a positive slow wave, and are often triggered by external stimuli. Sleep spindles are waxing and waning oscillations at around 12 to 15 Hz that last about 1 second and occur 5 to 10 times a minute. Eye movements and muscle tone are much reduced. At a behavior level, stage N2 qualifies fully as sleep because people are partially disconnected from the environment, meaning that they do not respond to the events around them. Their arousal threshold is increased. If stimuli are strong enough to wake them up, people in stage N2 will generally confirm that they were asleep. Non-REM sleep, stage 3. Stage N2 is followed, especially at the beginning of the night, by a period called Stage N3, during which the EEG shows prominent slow waves in the delta range, 0.5 to 2 Hz, more than 75 microvolt in humans. Eye movements cease during stage N3, and EMG activity decreases further. Stage N3 is also referred to as slow wave sleep, delta sleep, or deep sleep, since the threshold for arousal is higher than in stage N2. The process of awakening from slow wave sleep is drawn out, and subjects often remain confused for some time. REM sleep. After deepening through stages N2 and 3, non-REM sleep lightens and returns to stage N2, after which the sleeper enters REM sleep, also referred to as paradoxical sleep, because the EEG during REM sleep is similar to the activated EEG of waking or of stage N1. Indeed, the EEG of REM sleep is characterized by low voltage, fast activity, often with increased power in the theta band, 3 to 7 Hz. REM sleep is not subdivided into stages, but is rather described in terms of distinct tonic and phasic components. Tonic aspects are present throughout REM sleep. They include the activated EEG and a generalized loss of muscle tone, except for the extraocular and middle ear muscles and the diaphragm. Penile erections and pupillary constriction as a reflection of a parasympathetic dominance. Phasic features which occur episodically during REM sleep include irregular bursts of REM and brief contractions of middle ear and other muscles, twitches. Behaviorally, REM sleep is deep sleep, with an arousal threshold that is as high as in slow wave sleep. The sleep cycle. The succession of non-REM sleep stages followed by an episode of REM sleep is called a sleep cycle and lasts approximately 90 to 110 minutes in humans It has the scent of apple pie, which is my favorite scent out of all the candles that I have, and believe me, I have lots of them. So, sleep during the lifespan. Sleep patterns change markedly across the lifespan. Newborn infants spend 16 to 18 hours per day sleeping, with an early version of REM sleep called active sleep occupying about half of their sleep time. At approximately 3-4 months of age, when sleep starts to become consolidated during the sleep. The sleep age shows more mature waveforms and characteristic of non-REM and REM sleep. During early childhood, total sleep time decreases, and the REM sleep proportion drops to adult levels. The proportion of non-REM sleep spent in slow wave sleep increases during the first year of life, reaches a peak, declines during adolescence and adulthood, and may disappear entirely by age 60. The next chapter here is about brain centers regulating wakefulness and sleep. Things are going to get very biologic. Let's begin. Wakefulness system. Maintenance of wakefulness is dependent on several heterogeneous cell groups extending from the upper pons and midbrain, the so-called reticular activating system, RAS. glutamatergic cells, many of them glutamatergic, in the basal forebrain, which in turn provide an excitatory input to the entire cortex. Cholinergic neurons in the pons also project to the posterior hypothalamus, where histaminergic neurons are located in the tuberomammillary nucleus. Histaminergic neurons, which project throughout the cortex, fire at the highest rates during wakefulness and are inhibited during both non-REM and REM sleep. Probably the largest contingent of the wakefulness promoting system is made up by cells dispersed throughout the brainstem reticular formation and the basal forebrain that do not release conventional neuromediators, but rather the ubiquitous neurotransmitter glutamate. By binding to metabotropic receptors, glutamate can act as a neuromodulator and influence the excitability of target cells. The firing patterns of these glutamatergic cells are not well characterized. Noradrenergic cells are concentrated in the locus coeruleus, in the upper pons, from where they project throughout the brain. They fire tonically during wakefulness and emit short, phasic bursts of activity during behavioral choices or salient events. By contrast, locus coeruleus neurons decrease their firing during non-REM sleep and cease firing altogether during REM sleep. Serotoninergic cells from the dorsal raphe nucleus also project widely throughout the brain and, like noradrenergic cells, fire at higher levels in waking, lower levels in non-REM sleep, and fall silent during REM sleep. However, in contrast to noradrenergic neurons, serotoninergic neurons are inactivated when animals make behavioral choices or orient to salient stimuli, and are activated during repetitive motor activity, such as locomoting, grooming, or feeding. Dopamine-containing neurons located in the substantia nigra and ventral tegmental area, which innervate the frontal cortex, basal forebrain, and limbic structures, do not appear to change their firing rate depending on behavioral state, though blocking dopamine reuptake is known to promote arousal. Finally, the peptide hypocretin, also known as orexin, is produced by cells of the anterior hypothalamus that provide excitatory input to all components of the waking system. onset, wakefulness promoting neuronal groups are actively inhibited by antagonistic neuronal populations located in the hypothalamus and basal forebrain. Decrease in levels of acetylcholine and other waking-promoting neuromodulators and neurotransmitters lead to the opening of leak potassium channels in cortical and thalamic neurons, which become hyperpolarized and begin oscillating at low frequencies. Cell groups scattered within the anterior hypothalamus, including the ventrolateral preoptic area and the median preoptic nucleus, as well as in the basal forebrain, are involved in the initiation and maintenance of sleep. These neurons tend to fire during sleep and stop firing during wakefulness. When they are active, many of them release GABA and the peptide galanin and inhibit most waking-promoting areas, including cholinergic, noradrenergic, histaminergic, hyperkeratinergic, and serotoninergic cells. In turn, the latter inhibit several sleep-promoting neuronal groups. This reciprocal inhibition provides state stability, in that each state reinforces itself as well as inhibits the opponent state. REM sleep generator. This consists of pontine cholinergic cell groups, LDT and PPT, that are part of the wakefulness system, and nearby cell groups in the medial pontine reticular formation and medulla. Lesions in these areas eliminate REM sleep without significantly disrupting non-REM sleep. Pontine cholinergic neurons produce AG activation by releasing acetylcholine to the thalamus and to cholinergic and glutamatergic basal forebrain neurons that in turn activate the limbic system and cortex. However, while during wakefulness, other waking-promoting neuronal groups, such as noradrenergic, histaminergic, hyperkeratinergic, and serotoninergic neurons, are also active. They are inhibited during REM sleep. Other REM-active neurons in the dorsal bones are responsible for the tonic inhibition of muscle tone during REM sleep. Finally, neurons in the medial pontine reticular formation fire in bursts and produce phasic events of REM sleep, such as REM and muscle twitches. Neural correlates of wakefulness and sleep. spontaneous neural activity. Wakefulness. The waking aging characterized by the presence of low voltage fast activity is known as activated because most cortical neurons are steadily depolarized, close to their firing threshold, and are thus ready to respond to the slightest change in their inputs. The readiness to respond of cortical and thalamic neurons enables fast and effective interactions among distributed regions of the thalamocortical system, resulting in a continuously changing sequence of specific firing patterns. Superimposed on the low voltage fast activity by background of wakefulness, one frequently observes rhythmic oscillatory episodes. Within the alpha, beta, and gamma range, which are usually localized to specific cortical areas, these waking rhythms are due to the activation of oscillatory mechanisms intrinsic to each cell, as well as to the entrainment of oscillatory circuits among excitatory and inhibitory neurons. Non-REM sleep. The EEG of non-REM sleep differs markedly from that of wakefulness because of the occurrence of slow waves, K-complexes, and sleep spindles, the opening of leakage potassium channels due to the reduced levels of acetylcholine and other neuromodulators, draws cortical and thalamic cells toward hyperpolarization and triggers a series of membrane currents that produce the slow oscillation. Interesting. As shown by intracellular recordings, the slow oscillation is made up by of a hyperpolarization phase or downstate, which lasts a few hundreds of milliseconds, and a slightly longer depolarization phase or upstate. The downstate is associated with the virtual absence of synaptic activity within cortical networks. During the upstate, by contrast, cortical cells fire at rates that are as high or even higher than those seen in waking, and may even show periods of fast oscillatory activity in the gamma range. The slow oscillation is found in virtually every cortical neuron and is synchronized across much of the cortical mantle by cortical and thalamocortical connections, which is why the EEG records high voltage, low frequency waves. Human EEG recordings using 256 channels have revealed that EEG slow waves behave as traveling waves that sweep across a large stage 2 and more than 10 times a minute in stage 3. Thus, a wave of depolarization and intense synaptic activity, followed by a wave of hyperpolarization and synaptic silence, sweeps across the brain more and more frequently, just as non-REM sleep becomes deeper. Studies using intracerebral EEG recordings in epileptic patients have demonstrated that, particularly toward the end of the night, the slow oscillation tends to occur locally, asynchronously across different brain regions. These studies also demonstrated that small amplitude, slow waves in the scalp EEG mostly represent local waves, occurring out of phase across different brain regions, as opposed to large amplitude global EEG slow waves that are in phase across different brain regions. K-complexes, which can be triggered by external stimuli and appear particularly prominent because they are not immediately preceded or followed by other slow waves, are mostly like the EEG correlate of such global slow oscillations due to the near-synchronous activation of the cortical mantle by the RAS. Sleep spindles occur during the depolarized phase of the slow oscillation and are generated in thalamic circuits as a consequence of cortical firing. When the cortex enters an up state, strong cortical firing excites GABAergic neurons in the reticular nucleus of the thalamus. These in turn strongly inhibit thalamocortical neurons, triggering intrinsic currents that produce a rebound burst of action potentials. These bursts percolate within local thalamo-reticular circuits and produce oscillatory firing at around 12 to 15 Hz. Thalamic spindle sequences reach back to the cortex and are globally synchronized by corticothalamic circuits. emission tomography or PET studies show that metabolic activity and blood flow are globally reduced in REM sleep. In non-REM sleep, compared to rest and wakefulness, during slow wave sleep, metabolic activity can be reduced by as much as 40%. Metabolic activity is mostly due to the energetic requirements of synaptic transmission, and its reduction during non-REM sleep is thus most likely due to the hyperpolarized phase of the slow oscillation, during which synaptic activity is essentially abolished. At a regional level, activation is especially reduced in the thalamus due to its profound hyperpolarization during non-REM sleep. In the cerebral cortex, activation is reduced in dorsolateral prefrontal cortex, orbitofrontal, and anterior cingulate cortex. This deactivation is to be expected given that slow waves are especially prominent in these areas. Parietal cortex, precuneus, and posterior cingulate cortex. Parietal cortex, precuneus, and posterior cingulate cortex, as well as medial temporal cortex, also show relative deactivations. As discussed in other chapters, the deactivation of thalamus and associated frontoparietal networks is seen in all other conditions characterized by reduced consciousness, such as coma, vegetative state, and anesthesia. By contrast, primary sensory cortices are not deactivated compared to rest and wakefulness. Basal ganglia and cerebellum are also deactivated, probably because of the reduced inflow from cortical areas. During REM sleep, absolute levels of blood flow and metabolic activity are high, reaching levels similar to those seen during wakefulness, as would be expected based on the tonic depolarization and high firing rates of neurons. There are, however, interesting regional differences. Some brain areas are more active in REM sleep than in wakefulness. For example, there is a strong activation of limbic areas, including the amygdala and the parahippocampal cortex. Cerebral cortical areas that receive strong inputs from the amygdala, such as the anterior cingulate and the parietal lobule, are also activated, as are extrastriate areas. By contrast, the rest of parietal cortex, precuneus, posterior cingulate cortex, and dorsolateral prefrontal cortex are relatively deactivated. As will be mentioned in the next section, these regional activations and inactivations are consistent with differences in mental state between sleep and wakefulness. Upon awakening, blood flow is rapidly re-established in brainstem and thalamus, as well as in the anterior cingulate cortex. However, it can take up to 20 minutes for blood flow to be responsiveness to stimuli. The most striking behavioral consequence of falling asleep is a progressive disconnection from the environment. The threshold for responding to peripheral stimuli gradually increases with the succession of non-REM sleep stages, N1 to N3, and remains high during REM sleep, since cortical neurons continue to fire actively during sleep. How does this disconnection come about? Non-REM sleep. Due to the progressive intermittent hyperpolarization of thalamocortical neurons, sensory stimuli that normally would be relayed to the cortex often fail to do so because they do not manage to fire thalamocortical cells. In addition, the rhythmic hyperpolarization during sleep spindles is especially effective in blocking incoming stimuli since it imposes an intrinsic oscillatory rhythm that effectively decouples inputs from outputs. Thus, the thalamic gate to the cerebral cortex is partially closed. However, sensory stimuli in various modalities can still elicit evoked potentials from the cerebral cortex, and neuroimaging studies have shown that primary cortical areas are still being activated, as suggested by studies using transcranial magnetic stimulation in conjunction with high-density EEG. It is likely that during non-REM sleep, the activation of primary sensory areas is not followed by the activation of higher-order areas because of a breakdown in cortical effective connectivity. REM sleep. With the transition from non-REM to REM sleep, neurons return to be steadily depolarized, much as they are during quiet wakefulness. Yet, sensory stimuli are still ignored, as if the brain was focusing on its internal activities rather than on the environment. Not unlike states of intense absorption. While the underlying mechanisms are not clear, the prefrontal and parietal cortical areas that are deactivated in REM sleep are important for directing and sustaining attention to sensory cortices. Sensory inputs reaching primary cortices would then find themselves to be systematically unattended. Specifically, the reduction of serotonin release during REM sleep may favor a dissociative hallucinogenic state, as seen with certain psychoactive compounds. In addition, low levels of noradrenaline may contribute to perceptual disconnection during REM sleep. Indeed, noradrenaline is known to bias behavioral orientation toward the external versus the internal environment, and insufficient noradrenergic suppression may account for anesthesia awareness, a condition in which individuals become connected and experience their actual surroundings during anesthesia. Other people of other possible mechanisms underlie In a recent single-case study, the subcortical connectivity of the left posterior cingulate cortex was disrupted using intraoperative electrostimulation, which repeatedly led to disconnection. The patient became behaviorally unresponsive and later reported dreamlike conscious experiences unrelated to his actual environment in the operating room, including experiencing the sun, finding himself on the beach, and being surrounded by a white landscape. Nevertheless, in contrast to a person in a coma or a vegetative state, a sleeping person can always be awakened if stimuli are strong enough or especially meaningful. For example, it is well known that the sound of one's name or the wailing of a baby are among the most effective signals for awakening. And the next big chapter is about consciousness in sleep and also dreaming. There are two main lessons to be learned from the study of consciousness in sleep. The first is that during certain phases of sleep, the level of consciousness can decrease and at times nearly vanish, despite the fact that neural activity in the thalamocortical system is relatively stable. The second is that during other phases of sleep, vivid conscious experience is possible despite the sensory and motor disconnection from the environment and the loss of self-reflective thoughts. Changes in the level of consciousness. Studying mental activity during sleep offers a unique opportunity to find out how changes in brain activity are associated with changes in consciousness. When REM sleep was discovered, it was immediately noticed that if subjects were awakened from that stage of sleep, and asked whether they had a dream, they would say so at least 80% of the time. Subjects invariably reported dreams that were vivid, with characteristically intricate plots and changes of scene. Awakeness from non-REM sleep instead yielded dreams 20% of the time or less. These findings led to the approximate equation of a physiological state, REM sleep, with a cognitive state, dreams. This equation was encouraged by the remarkable similarity between the age of REM sleep with that of wakefulness as opposed to that of non-REM sleep. It seemed natural to infer that the activated, low-voltage, fast activity EEG of waking and REM sleep would support vivid conscious experience, while the deactivated, high-voltage, slow activity EEG of non-REM sleep would not. However, later studies have shown that the relationship between consciousness and sleep stages is more complicated. By just changing the question from "tell me if you had a dream" to "tell me anything that was going through your mind just before you woke up," the percentages of recalls from non-REM sleep reaches as high as 60%. It is now clear that reports indicative of conscious experience, including dreamlike experiences, can be believe sleep is the only phase of adult life during which healthy human subjects may deny that they were experiencing anything at all. On the other hand, between 60% and 80% of the time, awakenings from non-REM sleep yield reports with experiential content. The length of non-REM reports is widely distributed. Their median length is similar to that of reports from sleep onset. However, there are many very short reports early in the night and much longer reports later in the night, considerably longer than those typically obtained at sleep onset or even during quiet wakefulness. Reports from non-REM sleep, especially early in the night, are often thought-like. For example, I kept thinking about my upcoming exam and about the subject matter that it will contain. Later in the night, they can be much more hallucinatory and, generally speaking, more dreamlike. REM sleep. Awakenings from REM sleep yield reports 80 to 90% of the time, a percentage similar to that obtained at sleep onset. Especially in the morning hours, the percentage is close to 100. Most REM reports have the characteristics of typical dreams, complex, temporally unfolding hallucinatory episodes that can be as vivid as waking experience. For example, as reported by Hobson in 2002, as the climbing party rounds the trail to the right, I am suddenly on a bicycle, which I steer through the group of climbers. It becomes clear that I make a complete circuit of the peak at this level by staying on the grass. There is, in fact, a manicured lawn surface continuing between the rocks and the cracks. Then the scene changes to Martha's Vineyard Island, though I was still on the same bicycle, and then to a shopping center, a restaurant, a dance, and a meeting of faculty colleagues. One of my colleagues' wives is seen as a blonde when in reality she is a brunette. The sense of movement, which is continuous, becomes particularly delightful when I become practically weightless and glide along a golf fairway. At the dance, there is a Baltic group wearing embroidered peasant garb and stomping the floor to a loud band. I can hear the drums especially. Remarkably, the median word count of REM sleep reports is even higher than that of wakefulness reports, whether quiet or active. This finding seems to fit with the notion that dreams are single-minded and thus less frequently interrupted by extraneous thoughts than waking consciousness. Also, the average length of REM reports increases with the duration of the REM sleep episode. By contrast, there is no such relationship for non-REM sleep reports. What are the processes underlying the systematic changes in the level of consciousness during different phases of sleep? At first, it was assumed that the fading of consciousness during certain phases of sleep was due to the brain shutting down. However, while metabolism is reduced, the thalamocortical system remains active, Even though the level of activations of cortical neurons during the upstate of non-REM slow oscillations is as high as in wakefulness and REM sleep, the upstate of non-REM sleep is intrinsically unstable, in that it is inexorably terminated by the occurrence of a downstate, a generalized, stereotypical cessation of activity that can last for a tenth of a second or more. The transition from up to down states appears to be due to depolarization-dependent potassium currents and to short-term synaptic depression, both of which increase with the amount of prior activation. Indeed, during non-REM sleep, the stimulation of cortical neurons typically precipitates a downstate, and even spontaneous activity cannot last for long before a downstate is triggered. From this perspective, the incidence of spontaneous slow waves can provide a telling indicator of the degree of stability in thalamocortical networks. Thus, during stage 1 and the transition between wakefulness and sleep, the cortex enjoys periods of activation that can last up to a minute before a large slow wave sweeps through, which is consistent with reports of short hallucinatory sequences from awakening. In stage 2, early in the night, the EEG is similar to that of stage 1, but the intervals between large slow waves are much shorter, on average 12 seconds. Accordingly, reports are not only short but also thought-like in character. In stage 2, later in the morning, the intervals between large slow waves are longer, and reports are correspondingly longer and more dreamlike. The hallmark of slow wave sleep, which is prevalent early in the night, are the large slow waves that sweep through the cortex more than 10 times a minute in stage 3, suggesting an extreme degree of bistability. Correspondingly, reports are usually of short duration and often thought-like. At times, no experiential content is reported. Recent EEG studies in which sleep consciousness was systematically assessed using serial awakenings paradigms confirmed that reports of unconsciousness were preceded by higher power in the delta range and a higher slow wave density compared to reports of wakefulness, of consciousness. In stark contrast, during REM sleep, which predominates later in the night, the EEG is tonically activated and there are no slow waves sweeping the cortex. Accordingly, REM reports are on average much longer and usually yield vivid, prototypical dreams. Why would the level of consciousness reflect the degree of bistability of thalamocortical networks? A possible answer is offered by the integrated information theory of consciousness, which states that the level or quantity of consciousness is given by a system's capacity to generate integrated information. According to the theory, the brain substrate of consciousness is a complex of neural elements within the thalamocortical system that has a large repertoire of available states. Information, yet cannot be decomposed into a collection of causally independent subsystems. Integration, in this view, integrated information would be high during wakefulness because thalamocortical networks have a large repertoire of global firing patterns that are continuously available on a background of tonic depolarization. During early non-REM sleep, by contrast, the ensuing bistability would reduce this global repertoire through two mechanisms. First, a local activation would cause a local downstate preventing effective interactions with other brain areas. As a consequence, the main thalamocortical complex would break down into causally independent modules. Loss of integration. Second, to the extent that global activation patterns can still occur, they too would be rapidly followed by a global stereotypical downstate, thereby greatly reducing the repertoire of available states. Loss of information. To test these predictions, it is not sufficient to observe activity levels or patterns of temporal correlations among distant brain regions, functional connectivity, but it is crucial to employ a perturbational or causal approach, effective connectivity. For this purpose, TMS evoked brain responses were recorded using a high-density EEG system to investigate to what extent cortical regions can interact causally, integration, and produce differentiated responses. Information. As shown, TMS applied to various cortical regions during wakefulness induced a sustained response made of changing patterns of activity. Specifically, a sequence of time-locked, high-frequency oscillations occurred in the first 100 milliseconds and was followed by a few slower components that persisted until 300 milliseconds. Source modeling revealed that the initial response to TMS was followed by spatially and temporally differentiated patterns of activation, presumably mediated by long-range, ipsilateral, and transcallosal connections. As soon as the subjects transitioned into stage 1, This finding indicates that during early non-REM sleep, when the level of consciousness is reduced, effective connectivity among cortical regions breaks down, implying a corresponding breakdown of cortical integration. In subsequent experiments, it was found that when applied to a median centroparietal region, each TMS pulse would trigger a full-fledged, high-amplitude slow wave that closely resembled spontaneous ones and that traveled through much of the cortex. Spatially, the TMS-evoked slow wave was associated with a broad and stereotypical response. Cortical currents spread like an oil spot from the stimulated side to the rest of the brain. The large negative peak evoked by the TMS pulse, corresponding to a global cortical downstate, demonstrates that during early non-REM sleep activation is inevitably followed by deactivation, suggesting that the repertoire of possible firing patterns is drastically reduced. Importantly, such stereotypical responses could be induced even when, for the preceding seconds, there were no slow waves in the spontaneous EEG, indicating that perturbations can reveal the potential stability of a system, irrespective of its observed state. By contrast, during REM sleep late in the night, when dreams become long and vivid, and the level of consciousness returns to levels close to those of wakefulness, the responses to TMS also recovers and comes to resemble more closely those observed during wakefulness. Evoked patterns of activity become more complex and spatially differentiated, as it is shown in this figure. Although some late components are still missing, altogether these TMS EEG measurements suggest that the sleeping brain, despite being active and reactive, changes dramatically in its capacity to generate integrated information. It either breaks down in causally independent modules, or it bursts into a global stereotypical response in line with the predictions of the integrated information theory. Importantly, the use of a perturbational approach reveals that during non-REM sleep, cortical circuits may be intrinsically bistable even during periods of stable ongoing EEG with no overt slow waves. Dreams, consciousness in the absence of sensory inputs, and self-reflection, such as striking as the near loss of consciousness during certain phases of sleep, is its remarkable preservation during other phases. This is especially true of REM sleep awakenings, which yield reports of vivid dreams in the majority of cases. Perhaps the most remarkable property of dreams is how similar they can be to waking consciousness, to the point that the dreamer may be uncertain whether he is awake or asleep. This means that the sleeping brain, disconnected from the real world, is capable of generating a also have sound, tactile feelings, smells and tastes, as well as pleasure and pain. The categories that are the stuff of dreams are the same as those that constitute the fabric of wakefulness. Objects, animals, people, faces, places, and so on. Dream experiences are not necessarily all vivid and perceptual. There are also faint ideas, such as in wakefulness and various kinds of thoughts. Dreams are also rich in emotion. In fact, emotions are often very intense, especially fear and anxiety. Here in speech or conversation is also extremely frequent, and speech patterns are as grammatically correct as in waking life. Finally, there is a good correlation between our waking and dreaming selves with respect to mood, imaginativeness, and predominant concerns. For example, people dream most often about the individuals and interests that preoccupy them in waking life, and they show aggression in dreams towards the same people with whom they are in conflict in waking life. Building on the cognitive model of Folks and on the work of Hall on content analysis of dreams, William Domhoff has attempted a synthesis that he calls the neurocognitive model of dreaming. Domhoff proposes that dreaming is what the mature brain does when, first, primary sensory cortices are relatively inactivated, thus enforcing a partial disconnection from the external world. Dorsolateral prefrontal cortices are relatively inactivated, thus reducing our ability to exercise reflection and decision making. And finally, a subsystem of brain regions comprising limbic and paralimbic structures, as well as several association areas at the temporoparieto-occipital junction, is at a sufficient level of activation. According to Domhoff's model, dreamlike experiences can occur not only in non-REM sleep, but also during wakefulness, provided sensory and prefrontal cortices are sufficiently quiet. Like Folks, Domhoff emphasizes that the dreaming subsystem, when activated, is drawing on memory, schemas, and general knowledge to produce a kind of dramatized version of the world, and that these dramatizations are an active act of imagination rather than a mere reaction to random activation. More specifically, Domhoff argues that the system of scripts and schemas activated in dreams is nothing else but the organizational basis for all human knowledge and beliefs. Basic level categories, which can be represented by a single image, reflect distinctions among types of animals, such as cats and dogs, types of social interactions, such as friendly and aggressive, or types of actions, such as walking and running. Spatial relations categories are, for example, up, down, in front of, and in back of. Finally, sensorimotor categories are based on experiences related to temperature, motion, and touch. The systematic occurrence of basic experiential categories in dreams is confirmed by the analysis analysis of thousands of dreams from all over the world according to the whole Van de Castle system. Dreams may also build upon figurative thinking, conceptual metaphors, metonymies, ironies, and conceptual blends. As pointed out by Lakoff and Johnson, hundreds of primary conceptual metaphors actually map common experiential categories. For example, basic experiences like forms and motion are used to understand more difficult concepts like friendship. They have a form relationship, and time. Time flies by. Just as in waking thought, figurative thinking may be used in dreams when it expresses a conception better and more succinctly than an experiential concept does. To this extent, some dreams may indeed be symbolic. Finally, based on content analysis, Tom Hoppe concludes that most dreams deal with personal concerns. Typical ones are being inappropriately dressed, being lost, or being late for an examination. Personal concerns are very stable over the years, as well as across cultures, which may explain why dreams' themes are stable across life, across individuals, and around the world. Such personal concerns are also the subject of recurrent dreams and of the repetitive nightmares experienced by people suffering from post-traumatic stress disorder, generally in stage 2. Curiously, personal concerns in dreams are often stuck in the past, in a way that fits with the persistence of negative memories stored in the amygdala and other limbic circuits that are part of the brain's fear system. Taken all together, these remarkable similarities between waking and dreaming consciousness suggest that conscious experiences in both wakefulness and sleep share similar anatomal functional substrates. This was recently illustrated by an fMRI study in which a machine learning algorithm was trained while subjects were watching natural images in form of movies. In the second part of the experiment, this algorithm was successfully used to decode the content of conscious experiences that occurred in the fallen asleep period. As an example, the algorithm correctly predicted that the subject was most likely seeing characters in the 30 seconds before the awakening. Indeed, when awakened, the subject reported, "What I was looking at was some kind of characters. There was something like a writing paper for composing an essay, and I was looking at the characters from the essay, or whatever it was." Another recent study in which brain activity was recorded at high density and subjects were awakened at pseudo-random intervals and retrospectively reported if they had been conscious during sleep, and what identified content-specific activations for faces, places, movement, and speech that closely matched those observed in wakefulness during perception of the same contents. Despite the similarities between waking and consciousness,